Thanks, Alex. So I think I have the nicest job of the entire day, which is to introduce early career researchers who are really producing some phenomenal research using this resource. So this is the second year we've done this competition, and it's really just one way of UK Biobank kind of recognising that it really is the students and the postdocs who are the super users of UK Biobank. They're the ones who get their hands dirty. And so this is one way of recognising all the fantastic research that's been done worldwide. So the competition was for early researchers to submit their research abstracts. Second year we've done it. It was a really difficult task, actually, just because of the sheer volume and the high quality of the research that came through. So we had a very difficult job of whittling it down to 20 abstracts. Those are presented in poster form, which I hope you managed to have a look at over the lunch. And we have 2 runners-up and a winner, all of which will present their research to you now. So the winner of this competition unfortunately can't be here today. So hopefully we can have a video of his abstract. So without further ado, I'd like to introduce remotely Dr. Patrick Turley from Massachusetts General Hospital and the Broad Institute, and the title of his talk is Education Can Reduce Health Differences Related to Genetic Risk of Obesity. So can we take it away?
Hello, my name is Patrick Turley. I'm a postdoc at Massachusetts General Hospital and the Broad Institute. I apologize that I can't be there in person today. I'm teaching a course in California, but I'm so flattered to have the opportunity to be able to present this work to you all. My paper is titled Education Can Reduce Health Differences Related to Genetic Risk of Obesity, and it's joint work with Silvio Barcelos and Leandro Carvalho. It's well understood that education and genetics can each play an independent role in a person's health, but in this paper we ask the question, does genetic makeup moderate the effect of education on health? Or equivalently, does education affect the association between genetic makeup and health? We do this by combining a natural experiment, the 1972 Raising of School Leaving Age, or ROSLA, with polygenic scores in the UK Biobank. The results that I'll show you today are based on an analysis plan that was pre-registered before the full release of the UK Biobank genetic data. So let me start by describing the natural experiment. In 1972, England, Scotland, and Wales increased the minimum age at which a child could drop out of school from 15 to 16. As a result, there was a discontinuous increase in the educational attainment from one birth cohort to the next. We estimate in the UK Biobank sample that 14% of students received an extra year of schooling as a result of this reform. Students born just before and just after the birth date cutoff for this reform should be similar, except that one group was required to stay in school for longer. As a result, any differences in health between the two groups must be a result of the additional schooling. We can use a regression discontinuity design to estimate the causal effect of this additional year of school at age 15 on a person's health. Due to limited time, I'll only be able to discuss the effect of education on an index of body size, made up of several obesity-related measures like BMI, waist-hip ratio, and body fat percentage. We transform this index into a binary variable that roughly corresponds to the BMI threshold for obesity. As our measure of genetic risk, we use a BMI polygenic score, which uses hundreds of thousands of genetic markers to predict risk of obesity. Complete results for our lung health and blood pressure outcomes and for our educational attainment polygenic score can be found in our paper if you're interested. In order to better understand the baseline health conditions of those affected by the ROSLA, I've plotted here the fraction of individuals who have a body size index above our established threshold. For this figure, we restrict our sample to those born within a year before the ROSLA birth date cutoff and who dropped out of school at age 15. So these are all students who would have been forced to stay in school an extra year if they had been born just a year later. We also divide these individuals into 3 groups based on their BMI polygenic score. Unsurprisingly, we see here that those in the low polygenic score group have a much lower risk of obesity than those in the high polygenic score group. The difference in risk between these 2 groups is approximately 20 percentage points. In this figure, we show the estimated effect of an additional year of education at age 15 on body size for each of these groups again. Here we see little to no effect of education on the low BMI polygenic score group, but a substantial decrease in risk for the high polygenic score group. The difference in the effect between the low and high group is roughly 14 percentage points. Recall that the risk difference between these groups was 20 percentage points before the reform. This means that this 1 year of education reduced the health differences between these groups by nearly 75%. In conclusion, we see that education can have a substantial impact of improving rates of obesity, and that these effects are concentrated among those with the greatest genetic risk. These results challenge the notion of genetic determinism. Even though polygenic scores can explain a substantial amount of variation for some phenotype, social policies can still mitigate health differences that arise from genetic risk. I'd like to thank the many people who provided useful feedback and research assistance on this project. Also thanks to our funders who supported this work. And last, but certainly not least, a very special thanks to the UK Biobank for providing such a valuable data resource to the entire research community and for organizing this meeting today.
So we liked that one particularly because it made us think that, it's not all in our genes, and that, telling my kids that to do well at school is actually pretty important. Okay, so the runner-up, and I'd love to introduce, is Jiwing He from Harvard Medical School, who I'm very pleased to say could be here with us today. And she's going to talk about the systematic comparison of environmental exposures and polygenic scores to identify individuals at risk for common diseases in the UK. So can I welcome you onto the stage? And I'd also like you to present you with some prize money. Perhaps I should give this to you after the talk. Okay. Take it and run. Okay, thanks very much, Ewan.
Hi everyone, my name's Ishwan. I'm a first-year PhD student at Harvard Medical School. Thank you guys so much for giving me the opportunity to talk today. So today I wanted to talk about how G and X play a role in predicting your disease. So as we've learned in biology classes, P is equal to G plus X, where G is the genetic variance, your genetic predisposition, and then X is different environmental exposures. So these include pollution, your education, like we learned in the previous talk, your diet and exercise and things like that. And then your phenotype is obviously different diseases like type 2 diabetes, lung function, coronary artery disease, and AFib. So G has been very extensively studied, as we've seen throughout the talks. Genome-wide association studies, they are able to identify a lot of variants significantly associated with the disease. So as of May of this year, there's been over 138 associations found. However, the problem is a lot of these common diseases, they follow a common variant model. So this means that a lot of the common diseases are due to a lot of variants as compared to Mendelian diseases, or rare Mendelian diseases, which are usually due to a few variants that have very strong penetrance. So one recent method, as we've learned about, that have become increasingly popular is to look at polygenic risk scores. So this combines individual effects of basically all the variants in a genome-wide association study, and this also accounting for linkage equilibrium. And so this theoretically maximizes the power of GWAS studies, and so this would kind of help solve the problem of the common disease, common variant model. And so this is many of you are probably familiar with this study that was released that was published last year by Kara et al. And so they looked at several diseases, and what they were able to find was they were able to identify individuals who have greater than 3-fold increased risks in all these different diseases. And so on the x-axis here, they're showing the percentile of the polygenic risk score, and then the y-axis is within each percentile, what percent of those individuals have the disease of interest. So as you can see, the individuals in the last few percentiles have a much higher prevalence than those in the lower percentiles. However, there's not as many studies in the exposure side, and so I wanted to talk to you a little bit about how we can study maybe exposures more and why it's important. So I wanted to introduce you first to something known as an exposure-wide association study. So what you're doing here is it's very similar to a GWAS study, but instead of genetic variants on the x-axis, you're looking at different environmental exposures. So with a huge database like the UK Biobank, you're able to study hundreds of environmental exposure variants at the same time. So you can basically associate each of the environmental variants with the disease of interest and plot it in a similar Manhattan plot shown here. And the different colors would represent different categories of the environmental exposure. So you can see, so this is a figure shown for type 2 diabetes. So you can see some peaks for physical activity, alcohol, and smoking. So not very surprising. And then from there, what I wanted to do was actually calculate something similar to a polygenic risk score. And this, I termed this an exposure score, and basically it's calculated in a similar way as the polygenic risk score that we've been talking about in the past talks. And again, so here I'm showing the gradients for type 2 diabetes on the left, and then coronary artery disease on the right. And again, the x-axis is the percentile of the score, and then the y-axis is the prevalence within each of the percentiles. So as you can see, the exposure score is also able to do a good job of separating individuals who have higher at greater risk for these diseases. And so if you look at the last 10 percentile, for example, there's a much higher prevalence of the type 2 diabetes as well as coronary artery disease in those last 10 percentile compared to the rest of the population. So in conclusion, there's a lot of information in environmental exposures, so maybe we should try to incorporate these information into improving clinical care for these complex common diseases. And these large databases like the UK Biobank are able to allow us to study associations between the environment and phenotypes like we've never been able to before. So I'd like to thank everyone in the RAGU who's helped me with my work, as well as the different funders, and of course UK Biobank. If you have any questions, please reach out to me. Thank you.
Okay, and the final speaker, the final runner-up of this competition is Dr. Baptiste Couvé-Duchesne from University of Queensland in Brisbane. And he's going to talk about something a little bit different than polygenic risk scores, the widespread association between grey matter structure and the human phenome. Thanks very much.
Thank you very much for the opportunity to present today. The field of neuroimaging is at a turning point nowadays with following the recent availability of large databases, and the UK Biobank is probably the largest in the world. So I'm going to talk today about a few new analyses, or mostly new analyses, that we can do on brain images and that have been inspired by the field of genetics. So why getting the inspiration from genetics? And that's because studying the genome or studying brain images have things in common, and mostly the fact that we're dealing with really large number of measurements and really complex data sets. So the first concept that has been previously coined as morphometricity, and which I'm going to present today, is the total association between a phenotype and all the brain measurements that we can get from a brain image. And this is analogous to a SNP heritability, if you're familiar with the genetic world. Similarly, the genetic correlation, when we transpose SNPs into brain measurements, becomes the gray matter correlation that gives us an idea of whether the morphometricity of the traits are shared. And finally, the same way that we develop polygenic risk scores, we can start building gray matter scores that can help us predict from the brain images themselves. So we processed the images ourselves, combining the T1 and T2 weighted MRIs. And then we processed first the cortex using FreeSurfer, and the subcortical volumes using the Enigma protocols that are released, made freely available. And this led us with about 650,000 measurements per individual, mapping the cortical thickness and surface area, as well as the subcortical shape, or rather thickness again and area. So if you come back to the genetics, this is basically the size of a GWAS chip that we're now dealing with, but consisting of brain measurements. When we look at the brain morphometricity, hence the total association between phenotypes and the 600,000 brain measurements, these are the 58 phenotypes for which this association was significant in the UK Biobank. So this is on the first 10,000 image participants, and this is 58 phenotypes presented here out of a list of 175 that we selected from the UK Biobank. As you can see, there are large associations between age, sex, and body size, and the gray matter structure, which is in yellow in this circular bar plot. And we also found interesting associations between the gray matter shape, gray matter structure, and the maternal smoking around birth that you can see at the bottom there, but also with depression score and symptoms, substance use, alcohol and tobacco, as well as lifestyle measurements of activity levels, or even diet, self-reported diet questions. We looked at the replication in a subsequent 5,000 image participants of the UK Biobank. And as you can see in this XY plot, 23 out of 58 associations replicate in this smaller sample. And the point estimates were very similar across the replication and discovery samples. Now taking it to a bivariate level and looking at whether this morphometricity is unique to the traits or shared between pairs of traits. We estimated the gray matter correlation shown above the diagonal, and you can contrast that to residual correlations below the diagonal. So remember, we cannot infer any direction of effect here, because we're dealing with the brains and not with the genome anymore. So these can reflect causal, bidirectional relationships between the variable, and also all the associations with the brain could be causes or consequences of the phenotypes that we're studying. Yet we found a lot of large and significant gray matter correlations between some of the traits reported and measures of body size, such as BMI or body fat percentage. So this means that the morphometricity that we identified is not necessarily unique, but rather shared between some of the traits. And for the case of depression, for example, or diet or activity levels, the signal that we detect, the association we detect with the gray matter structure comes from the from the covariance of those phenotypes with body size. This is quite interesting because it can help us revisit some of the brain markers that have been identified for depression, for example, so further characterize them. But it also opens the way for transfer learning, which is a way of studying one phenotype by studying instead a related phenotype. I'll show an example in a minute. And then finally, to conclude on the prediction level, then we built gray matter scores, so weighting each individual vertex of the brain and combining them, summing this risk across the whole brain. And then we could make significant prediction for 56 out of these 58 phenotypes using a tenfold cross-validation on the discovery cohort. 42 of these scores were predictive in the UK Biobank replication sample and could even predict some phenotypes in the Human Connectome Project sample, which is much smaller and composed of younger adults from the US. So as you can see, the prediction varies greatly, and it is relatively good for age, with a prediction R-squares of almost 0.6, and it's much lower for the phenotypes that show lower morphometricity. What was interesting, coming back to transfer learning, is that we actually do better at predicting depression score by using a gray matter scores for BMI, rather than using a specific gray matter scores for depression. And that's due to the shared signal that we identified previously. And I think our example also shows that you can now apply those scores to completely new cohorts and basically study phenotypes that have not been collected, such as maternal smoking around birth in the HCP that correlated with nicotine dependence at adult age. And I would like to finish by thanking all the people who made this study possible. So my lab heads and colleagues and co-authors in Brisbane and in Paris, and most importantly, all the UK Biobank participants and staff. Thank you for your attention.
So hopefully those three examples really do showcase the diversity of fantastic research that's been done using UK Biobank by talented researchers from all over the world. So it is fantastic to see it being used in this way. So if you haven't already done so, please take the opportunity in the tea break, which is now, to grab a cup of tea or coffee and have a look at the posters. I've got strict instructions to tell you to be back here for 3:30, so we've got 20 minutes for the next session. So thank you very much.